Well, thank you, Kuhn. And our last speaker of the morning is truly a pleasure to have one of the greatest drug metabolism and pharmacologists from former CTL laboratory and then AstraZeneca and now Imperial College, Dr. Ian Wilson. Thank you.
You have to be British to know how embarrassing that just was. So thank you very much for the introduction. I cannot possibly live up to that, so I won't even try. One of the really good things is, of course, being the last speaker, you've seen what everybody else has said, and you know that actually you have nothing new to say. So this will be some elements of revision. I also hope to show you just what a difficult field we're working in. So to those of you in the room, nice to see you. To those of you online, well, you know, please forgive me for being British and jet-lagged. So I will try and talk to you about the effects of pharmaceutical exposures. Now, I am actually much more interested in what the bugs do to the drugs than what the drugs do to the bugs. And I'm also interested really in pharmaceutical level exposures rather than environmental ones. So this talk has given me more problems in the last couple of months than any other I've done in the last decade. So if it is a little bit incoherent, I hope you will say charitably allow it to be the jet lag and possibly my age and nothing to do with the fact that there's not much to say, because at the moment there isn't. So here are some questions that I have for you and for me. And if you look at the first one, which is can pharmaceuticals at therapeutic doses affect the gut microbiome, does anybody in the room still have the opinion that they can't? Right? Everything that's led up to this morning tells you that they can. Are there consequences for the host? Probably. Is there evidence that environmental drug exposures act directly on the human microbiome? Because I think that is the question everybody would like to know the answer to. We'll see where we get to at the end of this talk. What about the impact on human microbiomes of agricultural drug use? Really good question, that. And one which I think is at the top of a lot of people's minds. And then there are indirect effects, which to me is, if I change the microbiome, what does that do to the host? So let's do some background, and some of this will be revision. So the gut microbiome provides the host with an important external organ, because your gut might be inside you, but in fact, it's outside you, we had this morning, with very significant metabolic capacity, and that's both for endogenous metabolism and for xenobiotics. And the microbiota that form it are not passengers, they're crew. This is a symbiotic relationship. I mean, Matt Rednebo might be a superhumanly lucky person, but he's also a super organism. You know, the consortium of thousands and thousands and thousands of bacterial genes that live in your gut are part of you. You know, if you want to be philosophical, as I have nothing much to say, being philosophical is perhaps a good idea. Next time you look in the mirror, remember that you're an ecology, not an individual, right? You're about maximum 10% human based on your human genome. And your human genome doesn't change in your lifetime, but your gut microbiome can. All right, so that's pretty important. And environmental factors can exert a major influence on the development and evolution of an individual microbiome. As we've heard this morning more eloquently than I can put it, initially the microbiome is derived from the mother, but exposures to chemicals, including drugs, and environmental microorganisms can modify it. And the presence of the gut microbiome has a direct effect on the host and changes the host metabolic phenotype. So the metabotype, the metabolome, all that sort of stuff is changed by your gut flora. And there's lots and lots and lots of things in addition to drugs that affect your microbiome. So that's age, genetics, sex, diet, disease, all this sort of stuff, where you live, the built environment, the agricultural environment. It's wonderfully complicated. I mean, you could spend 10 lifetimes looking at all of this. Unfortunately, I don't have 10 lifetimes. Right, so let's do a bit of chemistry. This is an Nmr spectrum. It's an Nmr spectrum of urine from a normal rat, probably about 8 weeks old. If you start off at the right-hand side and move along, you'll see nice big peaks going up. This is a really beautiful fingerprint because I could train you in about 20 minutes to read this, even if you're not an NMR spectroscopist. And I can tell you that's a normal rat. It isn't drinking alcohol. It's not taking acetaminophen. It's excreting quite a lot of citrate, succinate, alpha-ketoglutarate. That's the peaks at the far right. And at this end, there's quite a lot of aromatics, including this set of signals here, the big ones, and that's hippuric acid. And hippuric acid is one of those nice molecules that's part of your metabolism and the microbiome. So anybody drink coffee at the break? Right, so you've just delivered a whole load of caffeic acid, which is in caffeinated drinks, to your gut microbiome. And they'll be chewing it up fairly soon. And between you and your microbiome, you will be making benzoic acid. If you ate a lot of sausages this morning, there's probably benzoic acid in there as well as a preservative. Right? So with multiple dietary sources of benzoic acid, you then glycine conjugate it as the final step. It comes out in the urine, but that's a direct input into your body, and you are actually acting as a vast waste disposal system for your gut microbiome. So again, if we're going to be philosophical, who's in charge here? Do you exist for your microbiome, or does your microbiome exist for you? It's, depending on your mood, you know which. Right, so that's a normal animal. Let's look at an abnormal one. This is a rat that has no gut flora, or at least not many. Can anybody not see the difference? And it's all in the aromatics. Also, we have now got a massive signal for formic acid in the urine. Okay, but the aromatic region is barren. That means two things. Firstly, the rats, if we had been giving them caffeic acid, would just be getting rid of it in some other way. Probably nothing would be happening to it. All right, so this is really very important, and it's okay. So this is one of those digital questions. If you like, scientists are very good at asking digital questions. Is it this or is it that? Now I'm going to spend the next however long I get given before I'm dragged off. So I'm telling you, actually, it's much more complicated than that. But here's a nice digital question and a good digital answer. So one of the things that we can talk about is what's the effect of host genetics. We would expect, certainly with animals where we can really mess around with the genetics, that we might see microbiome changes as a result. Right? Much more difficult to, if you look at the diversity in this room, there won't be a single microbiome in the room that's the same as anybody else's. If you look at your histories and your genetics, trying to dissect out all of these epidemiological factors, it takes a lot of work in a human population, but you can get clues from working with animals. So we looked at the fecal microbiomes of a fair number of strains of rats, and we used the microbiologist tools of 16S and DGG and things. And look, pretty colors, aren't they? Tried to arrange the Nmr Spectrum, but didn't quite manage it. Each different set of colors represents a different strain of mice and a different microbiome. So there you go. Genetics is important, isn't it? Well, if you deconstruct the experiment a little and think about some of the things you've heard about transmission of microbiomes through the generations, these animals, unlike us, haven't been living together. They've been grown up with their mothers over generations in different parts of the facility. Okay, so as I tell my students, if you get the result you're expecting, you're not looking hard enough, right? You've probably done the wrong experiment. The next experiment we did was we said to ourselves, okay, what if you put different strains of mouse into the same mother with embryo transplanting? And we asked people around in the lab, and well, they could do it. It was very, very difficult, would be expensive. And then we rediscovered how mouse strains are shipped out from the manufacturers. And if you want a couple of strains of mouse, they come in a couple of mothers, but the strains are mixed in, and you sort them out when they pass. Once the mice have been born, you genetically type them. So we did that experiment. And what we find is actually what's really important is not the genetics of the mouse, but the fecal microbiome of the mother. All right. So this is, for me, beginning to get a little worrying, because this then made us ask the next set of questions, which we get onto in a second, which is, all right, Is it the fecal microbiome of the mother, or is it the environment that the young pups are born into that's important? Given that the fecal microbiome of the mother will have generated that environment, but again, you're trying to ask a digital question, but it's a rather complicated thing. Now, in terms of host metabolic phenotype, if you look at the little dotogram, the PCA on the left, on the right, you'll see that we also get, even though the microbiomes are the same, what we find is the metabolic phenotypes for males and females are different. All right, so this relationship between the microbiome and the phenotype and stuff is getting to be a rather more complicated than we would like in an ideal world. All right. Am I worrying anybody? Well, I kind of should be, because some of this stuff could be important. So, but for mice, the maternal environment is more important than strain, and gender is more important than strain for the metabolic phenotype. All right, let's talk about plasticity. This is an experiment that I always describe badly, so I can only suggest that you read the paper. But what we did was we know that the microbiome is plastic. So if you take a young mouse and move it from one environment to another environment, it will acquire the microbiome of that environment. If you leave it until it's physiologically mature and you move it, it'll keep the microbiome that it started off with. So what we have here is a load of blue dots at the bottom. That's a bunch of mice that aren't being moved. They're in one environment. Then we've got some red and green dots. If I remember it correctly, and forgive me if I haven't, I always get this one wrong. We have one environment where animals are being moved to, right? One group of young rats is moved at 4 weeks before the microbiome has properly settled, and the other group is moved at 8 weeks when it's mature. The group that moves at 4 weeks acquires the microbiome of the environment it's moved into. The group that's moved at 8 weeks doesn't. Okay? So there's another nice confounding factor for any experiments that you're doing in animals. And remember, experiments in animals are the easy ones because you think you can control everything, whereas experiments with humans, you know, are impossible. You know, rats and mice don't lie. Humans lie all the time. When you ask them, have you been drinking? Have you taken any drugs? You know, have you stuck strictly to the diet? People will say yes, especially if they're British, okay? But they will be lying. So we then asked ourselves also, people live in families, right? So part of your environment is the family that you live with. Now, rats and mice live in families too. They live in cages, and they are also wonderfully coprophagic, which means they share everything. So what we have here is different strains of Zucker rat. So ones that are going to be homozygously lean, heterozygously lean, and those that are destined to become fat by they have a defect in the leptin receptor. They just eat and eat and eat. Now they're all eating the same diet. We can't put them all in the same cage, but we have put them in 6 separate families at 4 weeks. All right. Now, one of the things that you see, if you look, is that if you look at a 5-week-old rat in the 3 groups, each cage has a similar pattern, a gross pattern of bacteria. And if you look at much more in much more depth at 16S RNA sequences, you'll see that animals within a cage are like each other, but between cages are different. You also see that at 14 weeks, they're not the same as they were at 5 weeks. And that goes for the 16 sRNA data as well. So in designing animal experiments to understand what's going on, you have to be very careful how you house your animals, right? So if you house a test and a control group in different cages, you will see different microbiomes. That's got nothing to do with necessarily with what you've done, but everything to do with what was in the cage to start off with. All right, so this is interesting, and it's going to make work for lots of postdocs and PhD students, but it is something that we have to understand if we're going to be sure that the results we generate are meaningful. Okay. So we then asked what happens when you give rats antibiotics. So we took 3 groups of rats, 6 control, 6 were treated with antibiotics for 8 weeks and 6 for 8 days, and 6 that were treated for 4 days and then allowed to recover. And what you get is, this is the control rats. Then we've got 2 groups of rats that are recovering, and at the far end we've got rats that are still suppressed with continuous antibiotic treatment. The easiest slide to look at is the PCA up in the top right-hand corner. The black dots are the control animals, which form a nice group over that way. The red dots are the continuously suppressed animals, so that's the extreme, what happens when you suppress stuff. Now, when we were putting the animals into recovery groups, you can't ethically leave rats on their own in cages. They're social animals, they get stressed. So in order to treat the rats reasonably well, we put them into 2 separate groups of 3. If you look up there, you'll see we have 2 separate groups of 3 because they're recolonizing in that new environment. And they're recolonizing differently, as you can see from the patterns. This is one of the recolonizing groups. That's the other one. So, all of this is really very, very difficult. And I have no particular answers to this, except to think very, very hard about your experiments. Consider co-housing, control and test in the same place so that they are exposed to each other's feces and things like that, so that they're sharing bacteria. If you really want to demonstrate that you're having an effect, remember that you are all free living. So you're interacting with the environment and with other people all of the time. So that's why life is more difficult than it should be, which is quite frustrating. So let's go on to this. Well, we've already seen that at therapeutic doses, you can have a significant effect on the microbiome with drugs. I mean, I've picked up 3 here. Antibiotics, they're obvious. I mean, they're designed to kill bacteria, right? And what you do to your microbiome is just a nasty bit of collateral damage. Think of it as carpet bombing. Proton pump inhibitors, well, their pharmacology changes the pH of the gut. You change the ecology. It's like chopping down all the trees. In a valley, it will change all sorts of metabolic flows. And we've already heard about the non-steroidals. They actually cause damage. If you damage the gut, you're changing the environment. So this is a no-brainer. For those of you that want more information, I put in some papers here which I didn't get in time to go into the documentation, but there are both short and long-term impacts on the gut microbiota of taking antibiotics at therapeutic doses. Similarly for PPIs, proton pump inhibitors. So there's enough literature out there to say that drug companies certainly need to assess the impact of their drugs on the gut microbiota and the long-term downstream consequences of that. On the other hand, if I'm dying of some really unpleasant bacterial infection and it's me or the gut microbiota, frankly, guys, I can get you recolonized anytime. Yeah. So, we have to keep this in a sensible context. Everybody's worried about dying. In the Western world, we're living longer than we've ever lived in any environment, okay, at the moment. So we do need to keep a sensible set of stuff on it. Right. So, yes, there must be. Any drug that acts directly on the gut microbiota or indirectly will have a long-term consequence. How we deal with that is an interesting question, but let's worry about that for therapeutic doses. I am, at the moment, somewhat less concerned about the subject of this meeting, which is environmental exposure to antibiotics. I'm not worried at all about environmental exposure to PPIs, because if we think about the doses of PPIs that you need to have a pharmacological effect on you that will affect the microbiome, If you're getting that from drinking water or eating food, then you're living too close to the chemical factory that's manufacturing them, and you shouldn't be drinking their wastewater. I mean, this is not likely to have a huge effect. Antibiotics, on the other hand, slightly more complicated, but I'm still not desperately worried. On the other hand, a clear and present obvious effector is agricultural use of antibiotics, because that can lead directly to resistant strains of bacteria. And the great thing about bacteria is, unlike most environmental toxins, they can reproduce themselves and they get around. They can be passed from hand to hand and from hand to mouth. I don't know whether you've seen or heard about it, but apparently about 2 or 3 minutes after you've shaken hands with somebody, you'll move your hand so that you can sniff it. Right, isn't that interesting? Probably much more interesting than anything I've said. No one now is going to be shaking hands with anybody, but apparently people have observed, scientists, so it must be true, have observed this. And it's probably a way of getting some more information about the person you just met. The UK has recently had what I think is a very interesting document that really has to be worth reading. It's a UK review on antimicrobial resistance, which is seen, everybody knows there are no good antibiotics left, and even the good ones that are left are being fed to animals as growth promoters. So we're killing ourselves so that we can eat. So this review, commissioned by the British Prime Minister, so it must be important, and hosted by the Wellcome Trust, is tasked with recommending by the summer of 2016 a comprehensive package of actions to tackle antimicrobial resistance globally. And you have to wish them the very best of luck, probably the very best of British luck. They reviewed the literature and they found that of the papers they examined, only 5% said, well, there's no problem with exposing animals to antibiotics to increase food production because there is no problem. 72% found evidence of a link of transmission of resistant bacteria to humans. US figures say 70% of the nearly 9 million kilograms of antibiotic use in the US is for agriculture, compared to 30% for humans. And of those, the FDA reckon that the 31 of the antibiotics used for this are deemed to be important for human health. I mean, if I was of a mind to be worried by things like this, I certainly would be. So, in the last few minutes, I'd just like to consider briefly, let's suppose that these antimicrobially resistant bacteria get out and into you and start, because of their rather better evolutionary selective pressure, begin to take over, especially if you're unfortunate enough to take antibiotics, so that you knock everything down and these hardened chaps are able to take over. How might they affect metabolism and toxicity of the therapeutic drugs that you're taking in your daily life, which I say for me is the most interesting? So we've already mentioned briefly this morning this, which is the bacterial production from tyrosine of paracresol. Now, paracresol is really quite interesting. I mean, it's presumably produced by the bacteria that produce it as an antibiotic to kill other bacteria, because it's really quite a nasty compound. You absorb it into your body, and the main thing that you do is you make cresol sulfate, and you excrete that in your urine. So again, you're a waste disposal system for something that you really don't want. And one of the microorganisms that's seen to be quite good at producing this stuff is paracresol. It's C. difficile, right? Not something you want, but if you've got it, it also produces something else that you don't want, which is something that steals sulfation. I mean, you can argue that sulfation of phenols is probably something that has evolved to deal with the production of stuff like this. I mean, you don't produce paracresol yourself. It's not in your genome. It's definitely imported. Now, the trouble with sulfation is that it is highly specific for phenols, but limited in capacity. If you think about acetaminophen, it is another phenol. It's also a liver toxin, and is one of the major causes of liver transplantation in the US and in other developed communities because of its toxicity. And it kind of works like this. At a therapeutic dose, you'll put most of it down glucuronidation and sulfation. Initially, sulfation is preferred because it's more specific, but glucuronidation takes over when you run out of capacity. As you increase the dose, you begin to exceed the capacity of glucuronidation, which is a high capacity but low specificity pathway. And you then do something really stupid, which only organisms with CYP2E1 can do. You start making a reactive metabolite, which it's like pouring petrol onto a fire. It goes around, it alkylates proteins, it causes a lot of damage, all right? Sulfation is your first defense against acetaminophen. Now, you can imagine if you have a high load of C. diff that's producing paracresol, a lot of your preliminary defenses are down already. The next level of toxification you can think about is that paracresol can also be metabolized. And I see my light's going red. I have one slide left after this. I will be through it in a second. Paracresol is also metabolized to reactive metabolites. So you get a double whammy. So here are the concluding comments. You know, it's a forgotten organ, the microbiome. It's been forgotten every generation and then rediscovered by the next generation. I've discovered the microbiome twice in my scientific lifetime. This is much more interesting. You know, microbes produce waste products to be dealt with by the host, and these might well affect toxicity and efficacy. We are not yet in a position to say what environmental exposure to subtoxic concentrations of these drugs will do. And one of the problems that toxicologists have is dealing with low exposures over long periods, because in rats and things, that's quite expensive and no one wants to fund it, and it's quite difficult. The effects of colonizing your guts with antimicrobially resistant bugs, though, I think is much more obvious. And according to the New Scientist, that wonderful organ of general science, last month, the FDA reported sales of antibiotics approved for use in livestock rose 23% between 2009 and 2014. And the practice needs to end. The FDA suggested the end of the year as a deadline. You can again only wish them luck. So thank you for your attention. I hope some of the things I said were sufficiently amusing to have kept you awake.
Thank you very much. Please don't go away. I would like to announce we had a typo in the agenda. So unfortunately, for those of you who need coffee, you'll have to wait until 12:30. So we'll have a discussion at 12:30 and then lunch. So we'll break for lunch at 12:30 until 1:30. So we will not have a break immediately after this. So now we'll welcome questions. Can you use the microphone? Did you raise your hand?
Yeah. Yeah. I would have thought that announcement about lunch being delayed is enough to put anybody off asking questions. But.
Yeah. That was great, Ian. Thanks for the call to arms about antibiotic resistance. I wanted to make a quick comment about a recent paper. Unfortunately, I don't have my laptop with me, or I'd tell you a citation, but the antibiotic amounts in the environments like surface waters and stuff like that, even at locations close to CAFOs, are not necessarily selective in the sense that we define selectivity or susceptibility in the clinical laboratory, which has some correspondence to therapeutic ability. But the important point about this paper was that they showed that something that goes straight to the heart of the matter of that we don't have an antibiotic resistance problem. What we have is an antibiotic multi-resistance problem. That's why you can't treat with stuff that you used to be able to. So you only get multi with horizontal gene transfer. And so the heart of this paper was to show that even at levels of antibiotics 100-fold lower than what was known to be selective, plasmids persist more effectively in the laboratory. You grow a strain in the laboratory, it'll lose its plasmids, which is kind of the first observation of a real effect that I've seen on very, very low levels. So, yeah, thanks.
I think it was
a really useful comment, and I'd just like to respond to it by saying I'm not saying that there won't be effects of environmental exposure on humans. What I'm saying is it's going to be awfully, awfully difficult with all of the other things that we're seeing to actually prove it, which is tedious. It's nice to prove something like that.
Do you have another question? No? Well, I had a question on potential species differences, especially in studies of drugs in pharma. So in your experience, have you seen an attempt to perhaps explain a difference in either efficacy or toxicity across species by looking at the microbiome, whether, you know, from rat to mouse or from rat to human? That part of that may be metabolism, but maybe other factors as well. So do you have a case study in mind?
I can answer that one pretty easily, no. I don't think we've looked. Say that the microbiome is a relatively rediscovered organ for pharma, which has already enough problems of its own without having to worry about this. The only people I think who've been particularly concerned about this sort of thing have been people making antibacterials, which is the only place they've looked. I mean, certainly there will be differences in species that may well be trackable back down to the gut microbiota. So it's a really good question. One of the reasons I'm struggling to answer it is I've been thinking about it for a long time and not got a decent answer. And again, these will be quite expensive experiments to perform. And being able to distill out which is the host and which is the bug, or the bugs, is really difficult. Now, the example put up this morning about digoxin is a really nice one, because it is one particular bacterium, but it's also one particular mutant strain of that bacterium. So having that bacterium in your stomach doesn't mean that you'll reduce digoxin, but having that bacterium with the right strain. And that was one of the things that really messed up the early work, which was done in the 70s, which was tremendous to narrow it down to one species, but they couldn't get a real correlation in humans. And the reason they couldn't get a correlation in humans was it was often the wrong strain, right bug, wrong strain. And given that level of complexity, actually pinning something onto a drug and a host is quite difficult. Sorry, that's a really not very good answer. But it's a very good.
That shows the state of the art.
Yeah.
That could be a factor, but it really hasn't been researched. So thank you for that. Well, I'd like to thank the speakers of this morning and invite some of them to participate in the panel discussion. So Dr. Wilson, please don't go too far. Please take your seat. We'd like to invite Dr. Cerniglia and Drs. Patterson and Redinbo to join us, and Dr. Liu, please, as well.